The chromatin condenses and centrioles appear and move to the opposite sides of the poles and form spindle fibers. And the nuclear envelope breaks down and disappears. During metaphase, the second stage of mitosis, the spindle fibers fully develop and the chromosomes align at the center of the cell. In anaphase, the paired chromosomes or sister chromatids separate and move to the opposite poles of the cell. In the final stage, telophase, the cell separates into two daughter cells, the nuclear envelope reappears, in prophase 1, chromosome pairs with its homologue after it was duplicated and condensed. The homologue swap segments, this is called crossing over, and each chromosome becomes attached to microtubules of a newly formed spindle. In metaphase 1, chromosomes are centered in the middle of the cell. The sister chromatids of one homologue orient towards one pole, and those of the other homologue toward the opposite pole, and then the spindle is fully formed. In anaphase 1, the homologue chromosomes depart from each other. The sister chromatids of each chromosome remain attached, though. In telophase 1, the chromosomes depart to the opposite poles, the cytoplasm divides, and there are now two haploid cells. In the second stage of meiosis, prophase 2, the microtubules attach to the kinetochores of the already duplicated chromosomes. Then the chromosomes are moved towards the spindle's equator. Metaphase 2, all of the chromosomes are lined up at the spindle equator, midway between the poles. In anaphase 2, the chromatids separate and then they become independent chromosomes. The microtubules move to the separated chromosomes to the opposite poles. The chromosomes are now at the opposite ends of the cells. The nuclear envelope reforms around each set of chromosomes. Then the cytoplasm finally divides, and now there are 4 haploid cells.